Good evening, and welcome to this evening's Savor program. I'm Andrew Waterhouse, Director of the Robert Mondavi Institute for Wine and Food Science.
And I'm Mackenzie Smith, the University Librarian and Vice Provost for Digital Scholarship at UC Davis.
We created the Savor lecture series to bring UC Davis experts together with policymakers, industry, and thought leaders to discuss some of the biggest topics in wine and food research underway today at UC Davis. These talks contribute to the university's mission by engaging the public with that scholarship. This evening, we will explore how our sense of smell shapes our experience of food and the world around us. To open our program, I'm pleased to introduce Dean Helene Dillard, who leads the UC Davis College of Agricultural and Environmental Sciences. She's a strong advocate for extending our research to its users and the public, whether in ag, the government, or to nonprofits. She's also a strong supporter of experiential learning and encourages our professors to provide our students with real-world experience before they graduate. But she also appreciates the flavors and aromas of good food and wine. Welcome, Dean Dillard.
Thank you, Andy. On behalf of UC Davis, I'd like to welcome you all to tonight's Savor lecture. This topic is definitely timely. One of the more disturbing effects of COVID-19 is the loss of smell, and that has many of us thinking more deeply about how our sense of smell affects how we experience what we eat, what we drink, and how we engage with so many other aspects of our daily life. We've all experienced moments where a smell immediately takes us back to a moment in time, good or bad. These scents play a role in defining our life experiences, and COVID-19 makes the study of aroma that much more compelling. Tonight, we will dive into that topic deeper and hear about how smell and taste are closely linked. While this subject may be new for some of us, researchers at UC Davis have been studying aroma for many years. As home of the nation's leading programs in agriculture, wine, and food science, UC Davis has been exploring how we perceive the smell, taste, and texture of food for decades. This work began more than 70 years ago when Professor Dinsmore Webb and Professor Richard Kempner first began using the newly developed technique of gas chromatography to measure the aroma of compounds of wines and brandies. Professor Rosemarie Pangborn joined the Department of Food Science and Technology in the 60s and helped create sensory science as an academic field. And Professor Emerita Ann Noble, sensory scientist and flavor chemist for the Department of Viticulture and Enology, created the wine aroma wheel and was a mentor to the esteemed Professor Sue Ebeler, who will be speaking this evening. And speaking of Sue, I am so pleased to be introducing her to you all this evening. Sue's lab is focused on studying how agricultural practices, food processing, and storage all affect the aroma and flavors we perceive in what we eat and drink. But in addition to her critical work of finding answers to questions about flavors and educating the next generation of vintners and food scientists, she is also a tireless advocate for students and provides leadership for the College of Agricultural and Environmental Sciences on student support services, academic advising, recruitment, and outreach programs as the Associate Dean for Undergraduate Academic Programs. I am also pleased to introduce and welcome back to campus author Harold McGee. Harold writes about food science and cooking and has received numerous honors for his work. And for 5 years, he wrote a column, The Curious Cook, for the New York Times. He has contributed to the journal Nature, is a regular speaker at the Culinary Institute of America, and since 2010 has been a visiting lecturer at Harvard. He is best known for his James Beard Award-winning book on food and cooking, The Science and Lore of the Kitchen, initially published in 1984 and revised in 2004. So I am really looking forward to hearing from both Sue and Harold this evening. Without further ado, I'll hand it to you, Mackenzie, to get the program started.
Thank you, Helene. That was very kind of you. Tonight's discussion of how we perceive aromas and food in wine, aromas in food and wine around the world is one that I'm really looking forward to myself, since this subject has been a personal fascination of mine my whole life. My father had anosmia, so we spent many hours talking about smells, aromas, and how they can affect taste. As a neuropsychologist, he even studied the relationship between hunger and satiety with the consumption of foods that you can or cannot appreciate without a sense of smell. So this topic is very close to my heart. But before we dive into the topic of smell, I'd like to remind everyone that if you have any questions as you listen to the speakers' presentations, you can use the Q&A button at the bottom of your screen to submit those questions at any time. We'll take as many as we can during the Q&A period. To lead off the conversation, it's my great pleasure to introduce Professor of Viticulture and Enology, Sue Ebeler. As Dean Dillard said, her research focuses on the chemical makeup of grapes and wine and other foods and beverages, and how those chemical compounds, from aromas to antioxidants, can affect the flavor, quality, and health effects of what we eat. As Dean Dillard also mentioned, Sue is the Associate Dean for Undergraduate Academic Programs at the College of Agriculture and Environmental Sciences. And in that capacity, she's been a terrific partner with the library to design freshman seminars on how to do college-level library research. It so happens that Professor Ebeler is also the person who first brought Harold McGee to UC Davis more than 15 years ago. So it is particularly appropriate that they will be sharing the stage tonight. Sue, over to you.
Thank you so much, Mackenzie and Dean Dillard, for that great introduction. It's really nice to be here tonight, and I'm really looking forward to the conversation and the questions. So I wanted to put my presentation tonight sort of in the context of some of the things that Harold talks about in his book. And in his book, he talks about perceiving the aromas of the world around us. And he talks about a lot of the molecules that make up these aromas. And so I wanted to talk about how, as an analytical chemist and a flavor chemist, aroma chemist, we start to deconstruct these complex aromas into these individual aroma compounds. It's really challenging, though, to then reconstruct those aromas from the chemical composition because of complex interactions and perceptual changes that can occur that I'll also talk about. So trying to put this deconstruction, reconstruction context into the framework of perception of aroma. So we can think about the complex aromas that we perceive as mixtures of individual compounds. And most of what we perceive is really a very complex mixture. And we can think about the analogy to light, where we have white light, which is really a combination, a mixture of different wavelengths. It's not a perfect analogy because our perception and the perceptual processes are a little bit different for vision and olfaction. But similar to being able to separate out white light using a tool like a prism into the individual components, we have a tool as an analytical chemist where we can separate this complex mixture into individual components. And that tool for deconstructing an aroma is what we call a gas chromatograph, or the process is called gas chromatography. And with gas chromatography, what we have is it's basically an oven, a box that we can heat. And inside this oven, we have a column. And the column is very narrow, usually a few millimeters in diameter, and pretty long, usually 5, 10, sometimes even 100 meters long. And we place this complex mixture of molecules onto the column and start to heat up the oven. And as the oven heats up, the compounds start to volatilize based on their boiling point and their chemistry. And so as the oven heats up, different compounds start to volatilize over time, and we start to separate this mixture into the individual compounds. As the individual compounds come off the column, they go to a detector. And I'm showing here a mass spectrometer as a detector. And here the mass spectrometer is basically measuring the mass of, or a relative mass of, individual compounds. And we can start to identify the compounds then based on how long it takes to come off the column as well as their mass. This is converted into a profile. We'd see it on a computer screen or print out on a piece of paper. That's our chromatogram. And each one of these peaks on the chromatogram on the x-axis at the bottom is time. So this is showing over time different compounds coming off the column. And the intensity, how big the peak is, tells us how much of that compound is there. So in this mixture, we have 5 compounds, with this compound being in the highest amount. The other thing we have to do is to be able to extract these compounds and get them onto the column. So there's many ways we can do this. One of the common techniques used now is called solid phase microextraction. And with solid phase microextraction, we put our sample into a container. Here it's shown as a liquid, say maybe it's a wine, where we have headspace here, a vapor phase above the liquid sample. And the volatile compounds start to partition, move into that vapor phase. We put the solid phase microextraction syringe into that headspace, and on the tip of that syringe is a polymer. And that polymer starts to attract the volatiles that are in that headspace and starts to concentrate them onto that polymer. So we start to pull compounds from the liquid phase, concentrate them onto this fiber, and after a certain amount of time, after we've extracted what we want from the headspace, we take the syringe out, inject it into our gas chromatograph. The fiber gets heated up quickly, and the volatiles go onto the column, and then we start the separation process. So we have an extraction process, an injection, separation, detection, and then an analysis where we can identify and quantify what's present. So what does that look like for a real sample? So this is what a gas chromatographic profile of a Cabernet Sauvignon wine would look like. And you can see that it's a complex mixture of hundreds of compounds. And the compounds are present in very high concentration. This is a peak that is very high concentration to very low concentrations. We have very small, low concentration compounds. So what are some of those compounds? In wine, we have compounds that are coming from grapes. We have fermentation compounds that are formed during fermentation. And then we have changes that occur during storage or from contact with oak or other processing aids. So using some of the language from Harold's book, some of the compounds are some sulfur compounds. These are what Harold talks about in the very beginning of his book, some of these primordial sulfur compounds, and they have kind of a rotten egg, sulfury kind of aroma. They're usually formed during the fermentation process, and they are present usually in low concentrations in wine. We also have some of this starter carbon compounds that Harold talks a lot about. And these that are shown here are 6 carbon containing compounds, alcohols and aldehydes. And these compounds have a grassy green character to them, and they're ubiquitous in many, many plants, many environmental components. They are in grapes, and they tend to decrease in concentration over time. I do want to point out that I'm showing some of these images and structures of compounds, not so that it's overwhelming and so that there's a lot of chemistry here, but really so that you can start to understand the complexity and the diverse nature of the compounds that are in wine. So in addition to the starter set of compounds in the grapes, we have some terpenes. These give a floral fruity character to the grapes. And we have a series of compounds that are actually present in the grapes, but they're at they're in a non-volatile form. So we don't smell them until the grapes are crushed, or we have enzymes and acids that start to react with these compounds and form the volatile compound. So one example here is ionone, which has a very violet type character, damascanone, which has kind of an apple-like aroma, and TDN, which is in Cabernet Sauvignon, usually at very, very low levels, but is a very characteristic and desirable aroma of aged Riesling wines, and it gives a very kerosene WD-40 type of character. Also in the grapes is a pyrazine. Cabernet Sauvignon particularly has this nitrogen containing pyrazine, which gives a bell pepper character to the wines. And then a couple of other compounds, these three right here, that are formed during fermentation. So they're microbially produced, an ester, an aldehyde, and an alcohol. The ester is very fruity, generic fruity kind of aroma. And the acetaldehyde and phenylethanol have a rose-like character or a honey-like character. So very diverse compounds. No single one smells like wine, but all together we have this odor object or this perception of wine, along with all the other compounds that I haven't mentioned. So once we've deconstructed this aroma and we can start to identify what these individual compounds are, then what happens when we start to reconstruct, put them back together? What are the compounds that are really important to the overall aroma properties of a food or beverage or the environment around us? So it's really difficult, it turns out, to actually reconstruct aromas from the chemical composition alone. And that's because we have a lot of interactions that occur that add to the complexity of the aroma. And I'm going to talk about three types of interactions here, some that we've studied: masking, suppression, and additive effects, some qualitative changes that occur, and some matrix effects. So the first is looking at some masking and additive interactions, and mostly we're going to focus on a masking effect. So in this image, where it's basically, we can think about it like a sensory map of a series of wines. And what we started with was a very neutral base wine, didn't have a whole lot of character. And if we were to put that base wine on this graph, it's not shown here, but if we were to put it on this graph, it would be right in the middle, not a lot of character to it at all. And then we took that base wine and we added some natural bell pepper aroma in two different concentrations, a high bell pepper and a low bell pepper. And we had sensory judges evaluate this for bell pepper character, as well as, we'll talk about in a minute, some fruity types of characters, cherry and jammy and berry kinds of aromas. And so when the judges smelled this wine that had added bell pepper in it, the bell pepper intensity increased. Now, this wasn't a hugely overpowering bell pepper aroma, but just enough that the judges could say, oh yeah, there's a sense of bell pepper in there. And we actually did it at two different concentrations, a high and a low, and the judges can tell the difference between that high and low. Then we took that high bell pepper aroma wine and started to add in some fruity aromas, a raspberry or a strawberry or a generic berry. The bell pepper composition, the concentration didn't change at all. But as we added these fruity aromas, the perception of bell pepper starts to move on this map over to the right. And the right is where we perceive more floral and fruity kinds of characters. And so the raspberry, strawberry, berry aromas are masking the overall bell pepper character. The bell pepper concentration hasn't changed, but our perception of it changes as other compounds get added in. We also did this with some mint aromas, as well as a cooked vegetal corn kind of character, and added in the fruity characters. And in each case, you can see the base, the mint, and the cooked corn. As we add in fruity characters, the sensory space kind of moves to the right, to the more floral. We don't have as much masking effect as what we see with the bell pepper, but we see it in all of these cases. So another thing that makes understanding composition and how it relates to perception very challenging is that we can have qualitative changes in aromas as we mix two or more compounds together. And an example here is ethyl butyrate, which is an ester. It's in grapes, it's in apples, it's in lots of fruits, and it has a pretty generic fruity kind of aroma. And when we mix that with diacetyl, which is a compound that's the bell pepper, or sorry, it's the microwave popcorn butter kind of aroma, buttery aroma. When we mix those two together, it doesn't smell like fruit, it doesn't smell like butter anymore. It smells totally different. It's a butterscotch kind of aroma, a completely new aroma that we perceive. And there's no way we could have really predicted what that new aroma would be from the individual compounds, their structures, or their aroma properties alone. So how can we start to understand some of these aroma interactions? Well, we can do all kinds of combinations individually if we know what the compounds are and we know their concentrations, but oftentimes we don't and don't have that information. And so what we developed in my lab is an instrument where we have a gas chromatograph again. Here's at the left-hand side is our GC instrument again, where we are separating the compounds of a mixture into the individual components. And as they come off the column, we can split them and go again to a detector and mass spectrometer. But we can also have at the end of the column a detector, which is a human nose. So a human is sitting there and smelling these compounds individually as they come off, describing them and saying, oh, that smells like coconut, that smells like butter. And so we can start to get qualitative aromas from individual compounds. So that's called GC, gas chromatography olfactometry. And that's a pretty common technique. What we did is, as these compounds are coming off of the column, we did two things. We put a switch in here so that we can take out an individual compound or a group of compounds and start to understand what does one compound or two or more compounds, how is that, if we remove that, how is that going to impact the aroma? And then we also, right before the olfactory port where the person is smelling, we put a cryo trap. And so we separate these compounds, remove a few of them, and then they come into this cryo trap, which is at liquid nitrogen temperature or maybe dry ice temperature. And the compounds recondense back together. So they recompose all back together, and we can smell them again as a mixture with one or a group of compounds removed. So what does that look like? This is an experiment we did with lavender, with fresh lavender from the gardens at the Robert Mondavi Institute. And we used our GC recomposition olfactometer, and you can see in the background, this is our GC trace, lots of compounds that are there. The interesting thing about fresh lavender is it's really hard to know what are the compounds that really contribute to the overall lavender character. It's a very complex aroma, and there's not one or just a few compounds that contribute to fresh lavender aroma. So we extracted the volatiles using solid phase microextraction, like I showed you, of fresh lavender, and we injected them, and then we recombined everything together in this top row and smelled it back reconstituted as a mixture. And we have this fresh lavender, dried lavender character, and the judges said that that reconstitution mixture smelled very much like the original. So we have a proof of concept that we can separate these compounds, bring them back together, and it smells a lot like the original. Then what the switching valve allows us to do is a series of experiments where we can, so in the 01 experiment to the just below the top, we've removed the compounds on the left-hand side of the chromatogram, and we've recombined the compounds in green on the right-hand side of the chromatogram. And we still have this nice fresh lavender character. So the compounds that combine together to give a fresh lavender character are the ones in this later half of the chromatogram. But then in 02, the next one down, we've removed part of the middle part of the chromatogram and smelled all the other compounds together. And now all of a sudden we've lost the fresh lavender character. We still have some citrus, hay, floral, root beer kinds of character, but no longer fresh lavender. So there's something about the mixture of compounds here in the middle that's really critical for creating this overall perception of lavender. And we can continue doing this all the way down till we can start to identify what is that critical mixture of compounds that gives us this overall fresh lavender character. I haven't shown it here, but we could continue these recomposition experiments. But it gives you an idea of how the complexity of what it takes to really understand some of the drivers of the overall aroma and how these compounds are interacting together. The last thing I'll talk about are matrix effects. And here what we're talking about is in a food system, usually it's not just water and aroma compound. we usually have lipids or fats that are present. We might have proteins. In the case of a wine, we have phenolic compounds. And in most of those cases, those compounds don't have a smell of their own. They're non-volatile, but they can affect the volatility, the release of the aroma compounds. And so that's what I'm showing here. We have these lipid globules or micelles here that are in an aqueous matrix. This would be like a salad dressing maybe, or we could think about ice cream that has a lot of fat in it. And the volatile compounds here, the smaller compound, these small orange dots, this is a volatile compound. They oftentimes like to be in a more fatty kind of a phase. And so they will solubilize in this lipid phase. And then when they're in this lipid phase, they're less likely to volatilize and be in the vapor so that we can perceive them. So as we change the matrix composition, add more fat, the volatile compounds will start to solubilize, partition into this fat lipid phase, and their concentration in the vapor phase will decrease. And so our perception of them decreases. So for example, a full fat and a low fat ice cream, even if we have the exact same aroma composition, the aroma perception will be very different because the flavor compounds are solubilized and sort of stored in these fat particles. So it adds, again, further to the complexity of what we're smelling. So with that, I'm going to end and just sort of indicate again that, you know, there's, as an analytical chemist, a flavor chemist, we have tools that we can start to deconstruct, identify, and quantify the aroma compounds that are present in our food, beverage, the world around us. But the complexity of trying to put these back together and trying to understand how, as humans, we perceive this complex mixture, there still is so much that we don't know and a lot that we still need to research and study. And so with that, I'm going to turn it back over to Mackenzie.
Thanks, Sue, for that fantastic overview of the science of aromas and how they interact. Very interesting. So now we turn to how that science is embodied in the world around us. As Dean Dillard said, Harold McGee is a world-renowned author on the subject of the science of cooking and food, and it's hard to describe how important Harold's work has been to the food scene over the last few decades, going far beyond his best-known books and articles and into influential niches of the modern food scene, like Lucky Peach magazine, one of my favorites. He is beloved by a generation of chefs and other people who are serious about food. Tonight, Harold will be discussing his new book, Nosedive, A Field Guide to the World's Smells, for which he spent 10 years in a deep dive into the world of aromas, the chemistry of smell, and sensory science. Harold, thank you so much for joining us tonight. Over to you.
All right. Thank you very much, Mackenzie. And I can't say how happy I am to be with all of you this evening, because I could not have written my books without UC Davis, and particularly without the library at UC Davis. So it's just a tremendous pleasure to be with all of you. And there is an organization, the Robert Mondavi Institute, that helps disseminate the work that's done at places like, well, at UC Davis in particular. And they have a Friends program. I'm a friend, and there are hundreds of people watching this evening. So I hope some of you anyway will consider joining the Robert Mondavi Institute as a friend and help support the kind of research that you heard Sue talk about and the kind of outreach that made it possible for me to write my books. Anyhow, so I've been writing about food and cooking for a long time, for 40 years. And I've always been interested in flavor above all, because that's what gives us the pleasure of eating. And when I first started writing about food back in the 70s, we didn't know a whole lot. So I couldn't write very much about it. Couldn't really understand that much about it. Fast forward to the years that I spent at UC Davis doing the research for the revision of my book on food and cooking. back in the early 2000s, a lot of information had been developed, both what's out there in the world that gives us this kind of pleasure, and then what's happening in our minds, in our brains, in order to register what's out there. And so I decided, after finishing the revision of On Food and Cooking in 2004, that I would start writing about flavor. The time was ripe. Well, I had to take care of a couple of other things, so I didn't really get started until 2010. And then when I sat down and looked at the situation, it was pretty clear that there was just too much at that point to talk about. We knew so much about what's going on inside ourselves. We knew so much about what's going on in things like wine, thanks to the work of Sue and her colleagues, that I couldn't really cover both. And so I ended up focusing in particular on smells. And the reason for that is that, and smells of the world. So, you know, this is the book that I took 10 years to write. And it's not the smells of food, although smells of food are included. It's about the smells of the world. And the reason for that is twofold. First of all, smell is the sense that gives us the most variety in our experience of flavors. Taste gives us a half dozen different sensations, but smell gives us uncountable numbers of sensations. So that's really what gives us the sense for the diversity of the foods that are out there. And then it's also smell that's our connection. It connects our experience of foods with our experience of the outside world, because we detect smells when we breathe in, smells from the rest of the world, and we detect the aromas of foods when we breathe out. So we get information about both. And smells are also, in a way, the most direct contact we have with the physical world around us. If you think about it, sight and hearing are very indirect. We're registering photons or pressure waves. But in the case of smell, we're actually detecting little bits of the world or little bits of the food with receptors, and for a split second, actually registering what the material realness of that stuff is. So to me, that's just amazing and fascinating, and has been largely ignored by sensory psychologists for a long, long time. Smell is coming into its own these days for all kinds of reasons, and I think it's long overdue. Anyway, so I've written this book about the world's smells, and what I hope it'll do is encourage people to, the title of this lecture series is Savor. We're all used to savoring food and drink, but what I'd like to encourage is that we savor the rest of the world in the same way. That is to say, pay as much attention to our sensory encounters with the world as we do with our sensory encounters with food and drink. Anyway, I'd like to share with you a couple of slides. So I'm going to see if I can manage to do that. There we go. So for those of you who would like an idea of what's in the book without having to go off and purchase it, you can get an idea by looking at some excerpts that have been published recently. None of them, it turns out, have much to do with food. So the Wall Street Journal published an excerpt about the molecules that exist in interstellar space, which, if we were capable of living in interstellar space, we would recognize as smells from planet Earth. And the reason for that is that some volatile molecules, some smell molecules are primordial. They were formed long before there was a planet, long before there were fruits and vegetables and so on. And I think that gives us a really interesting perspective on smell in general. Then Wired magazine decided to do an excerpt from the book about excrement, of all things. Of course, if I'm going to cover the smells of the world, that's a pretty prominent one, so I couldn't ignore it. And it turned out that Wired found a great deal of interest in connecting our mastery of that material with modern technology. So that's another excerpt you can see. And then just last week, the New Yorker ran a review that focused on fragrances, perfumes, and body odors. And that's yet another aspect of the smells of the world that I did try to cover. Now, because this is the Robert Mondavi Institute for Wine and Food Science, I thought I would just tell you a couple of quick stories to do with wine and food, just to give you a whiff of what it is that I'm trying to do in exploring these aspects of food and drink. So let's begin with cooking and with food. And to me, there is no better example of the alchemy of cooking than what you see going on the surface of this creme brulee. That is to say, the bruleeing of the sugar. So for molecules to be smellable, they have to be small. They have to be volatile so they can fly through the air into our nose. And most materials on Earth are made up of big molecules, proteins and carbohydrates and fats and things like that. These things are not volatile. One of the things that cooking does to make food interesting is to break those large molecules apart into smaller ones, into fragments that we can actually detect. And that's where a lot of the aroma of foods come from. And there's no better illustration of this than the caramelization of sugar, which is what you see going on there. It turns out that, usually when cooks do caramelization, they do it very quickly. And so with very high heat. And so you see the sugar kind of melting as the heating goes on and the mixture begins to turn brown. But in fact, you can get the same effect by heating sugar crystals very, very slowly. So this is a picture of some sugar crystals that I kept in my oven for a few days at a pretty low temperature. I forget what exactly it was, maybe around 200, 250 degrees. And over the course of several days, the caramel develops and gets darker and darker and darker. And the amazing thing about this process, the reason I call it alchemical, is that you start with one molecule, which is illustrated there. It's a sucrose molecule, 12 carbons and some hydrogens and some oxygens, and that's about it. It's a pure material, that's all there is. And then all you have to do as a cook is add energy in the form of heat. And that energy is sufficient to break that molecule apart into smaller molecules that do have aromas. Of course, sucrose, table sugar, has no aroma whatsoever. And so you do get a couple of molecules that are reminiscent on their own of caramel all by itself. But they're only the tip of the iceberg. It turns out that when flavor chemists have analyzed what it is that this process eventuates in, the number of volatile molecules that are produced, it's in the hundreds. And the most prominent ones are what you see here. So each of these molecules is going to provide a note either directly or, as Professor Ebeler said, by interaction with other molecules in the same mixture, notes of all sorts, from vinegar and buttery, that's diacetyl, which Professor Ebeler mentioned. But there are earthy, bready aromas. There's vanilla, the actual compound that gives us the smell of vanilla in vanilla beans is produced by caramelization. And the further you carry it, the darker and darker the caramel gets, the more you end up with molecules at the bottom of the list there, including cresol, the horse barn and tar aroma, and then smoky guaiacol. So you begin to get into the realm of not just caramelized, but scorched. Anyway, a tremendous diversity from a very, very simple action. And if you think about how complex fruits and vegetables and meats are to begin with, and then you apply very different kinds of heat, different levels and different periods of time, you can begin to appreciate just the remarkable diversity of molecules that we're detecting every time we have something edible. Now, switching gears to wine. And one of the first encounters I had with the Robert Mondavi Institute had to do with this terroir conference that took place in 2006. Terroir is something that has become a very exciting idea and the subject of lots and lots of speculation over the last decade or two. That is the idea that you can somehow experience the placeness, the whereness of a wine by experiencing its sensory characteristics. So the place somehow has a definitive and detectable influence on the final flavor of the wine. Lots and lots of work has been done on this. People have explored it in many different ways. One of those people is a famous graduate of the viticulture program at UC Davis, Randall Graham of the Bonnie Doon Winery, who's always doing wonderful experiments and thought-provoking, emotion-provoking demonstrations. So back in 2003, he astonished British wine journalists with his interpretation of this idea. And minerality is another term that's often used to give the sense that you're able to taste the mineral quality of different soils in different wines. And so he demonstrated this by infusing one of his wines with different rocks. That is to say, just putting rocks themselves into the barrels with the wines and letting the wines extract what they could. And they found that the flavors were, of course, very strongly affected by this technique, and including the control wine that had nothing added to it, they added up to a rock quartet. So this is something that is of great interest to a lot of people, has been for a long time. In 2007, I came up to UC Davis and interviewed a number of faculty members, Hildegard Heyman and Mark Matthew and a number of others, to figure out what it is that we could say at that point about how it is that the soil, the environment, the mineral content of the soil could actually affect the flavors that we detect in a glass of wine. And at the time, 2007, the answer seemed to be it would be very difficult for there to be a direct connection between the mineral content of a soil and the minerality, mineral flavors that we might think that we're detecting in a wine. And so Daniel Patterson, a chef, and I wrote this up for the New York Times. Well, science marches on, and we're always learning something new. And so I wanted to show you how it is that understanding something like the subject that Wired magazine was interested in, namely excrement, can end up being illuminating when it comes to our understanding of wine. So back in 2013, at Firmenich, which is a flavor and fragrance company based in Switzerland, people were doing work on the nature of the smells of latrines in order to come up with more effective ways of masking or overcoming those smells, controlling those smells, so that latrines would be more attractive for use in places where that was the only way to deal with human waste. And so flavor chemists at Firmenich analyzed the aromas of latrines, and they came up with, and using the same techniques that Professor Ebeler was talking about when it comes to analyzing wines, gas chromatography and so on, they found that this set of molecules was primarily responsible for the horrendous smell of latrines, including several sulfur-containing compounds. And I've shown at the right hydrogen sulfide, the simplest of these compounds, which they didn't even bother to include because it's so common. But that's another important part of it. Anyway, so they did that analysis, and then a few years later, they were following that up in their laboratory using a laboratory container containing hydrogen sulfide, pure hydrogen sulfide. And in that work, using that container, they started to smell things that weren't quite hydrogen sulfide. They were, trained noses, as they're called, so that they were able to tell that behind that hydrogen sulfide, there were some other things going on that shouldn't have been there. And so they followed their nose. They checked up on what was going on, and they found that the hydrogen sulfide had been contaminated by a few other trace compounds. In particular, these di and trisulfanes, very clearly closely related to hydrogen sulfide, but different. And the way that they noticed these compounds in the first place was that they began to smell things coming from that cylinder of hydrogen sulfide that reminded them of other things. Those other things included fireworks. They included the smell of a dentist drilling into your tooth, and they included the smell of smashing two pieces of flint together. And so they began to think, this is very interesting. I wonder if some of these molecules are involved in some of those other smells. Now, fireworks are difficult to do experiments on. Flint, not so difficult. And so they managed to design an experiment so that they could smash two pieces of flint together, take the headspace off, and analyze it for these very trace and reactive compounds. So it was quite a challenge analytically, but they were able to do it. And they were able to show that those molecules, in fact, are present when two pieces of flint are struck together. And of course, they're flavor and fragrance chemists. They're in Europe. They also thought about minerality. Perhaps this could be one of the keys to the experience of minerality in wines. And so they analyzed a local Swiss wine, Chasselas, which is often found to have mineral quality. And lo and behold, they found those same molecules in Chasselas, and they found higher levels of those compounds in wines that judges judged to be higher in mineral quality. So from latrines to minerality in wine, that's what paying attention to volatiles, the volatiles of the world, and not just food, not just wine, that's the kind of insight that kind of broad-ranging look can bring. And that's really what I'd like to encourage all of you to do, is simply to, whenever your attention is caught by something in the world, by something that you get a whiff of, to stop and savor it, to stop and smell not just roses and wines and creme brulees, but anything that might that you might add to your database of smell experiences, and have those experiences inform your experience of these more delicious things. And with that, I will turn it back to Mackenzie for the next step.
The next step. Thank you, Harold. That was fascinating and wonderful as always. I really appreciate that overview. And your book is amazing. I've read it myself and I highly recommend it to everybody here. So now we're going to turn to some Q&A, questions and answers, and we have about 20 minutes for that. So I'm going to start off with a fairly simple question for you, Harold, that kind of builds on what you were just saying, and that is, for those listeners who are interested in becoming more attuned to the smells around us, are there ways you can recommend to learn to deconstruct an aroma at home and start to train yourself a bit? How do you educate your nose?
Well, I guess the first thing I would say is that deconstruction can be a lot of fun, but it's going against the grain, because our one of the things we've learned over the last few decades is that our brains are designed to integrate information, not to pull it apart into its individual little bits. So it is going against the grain, and it does take some effort, but it can be fascinating, as wine lovers will attest. And it is something that you can learn. That's how perfumers and wine assessors and assessors and many others do it. And at home, one of the things that is very easy to do is to take a spice mixture, something like a curry powder, and take it off your spice shelf, and then put next to it some of the component spices that go into it. And you can, refer to the web or books to see what those might be. But, there's going to be cinnamon and ginger and garlic and various things. Turmeric is a big one. Smell the mixture and then smell the individual components, and then come back to the mixture and see if you can pick out that individual component. Something that's also fun, if you're not a big fan of curry, is to do that same kind of thing with a cola drink. And you can look online and see which essential oils go into the cola mixture, and then come back to your cola and see if you can pick those out, and maybe even try to make your own cola mixture using the different oils. So it can be a lot of fun to educate your nose that way.
Yeah, so something people can try at home for a change. Good. So kind of building on that, Sue, I have a question for you, which is, are some chemicals so pungent that even if they're in very small amounts, their presence would stand out more than some of the others in a mix like that?
Sure. So a couple of examples from the wine world. The cork taint aroma, trichloroanisole. Our human nose is very exquisitely sensitive to the TCA aroma, and it has this musty kind of aroma to it. And so it doesn't take very much to be there, and we can start to sense it. And it doesn't take very much to start to be overpowering. So we can start to pick out that musty aroma right away. And oftentimes when in food, in the beverage world, or in anything that sort of has an off aroma to it, it's often because the aroma compounds aren't in balance. In what we might use like an orchestra kind of term here. It's not really in balance. Not all of the molecules are interacting in sort of the normal way. One or more of them is in a higher concentration, and it starts to overpower the others, and then it doesn't smell right anymore. And so that can easily happen. And often as a perfumist, as a flavorist, as someone who studies wines and foods, and working with viticulturists and enologists to process and make food and wine, what we're trying to do is to create that balance so that there's not one compound or another that stands out.
Thank you. So building on that, I have a question for both of you, which is this notion that we are often told that there are correct pairings for food and wine in particular. And I was curious to know if how aroma would play into that, and if there's any scientific basis for that belief. Harold, why don't you start?
Okay. Well, that again is a subject that I've been interested in for a long time, and many people are interested in it and write books every year on the subject with new permutations and combinations, which I think maybe in and of itself is an indication that maybe there isn't a particular answer to this, because people keep coming up with new ways of presenting the same wines and the same foods. So when it comes to pairing and whether one thing goes with another, we're really talking about individual perception. We're not really talking about what's in the materials in an objective way. It's how an individual brain is perceiving that mixture. And different brains are going to have different reactions depending on whether they recognize those ingredients, whether they're liked or disliked for reasons that have nothing to do with their own, with the material's intrinsic qualities. There are just so many different variables that there are general guidelines that people have come up with in wine and food pairing that I think have more to do with taste and texture than with aroma. At least that's my sense.
Sue?
Yeah, what I generally tell people is to drink what you enjoy and drink it with what you enjoy, and try it out. Maybe it works, maybe it doesn't, but you know, it's an experience that you have and you learn from, and you find the things that you do like together. I guess I'll maybe sort of add something from Harold's book. I was looking at a few things in it tonight just before the talk. And he talked in one place about, I think it was fish and some metals that are in certain kinds of fish, that you then have a residue of that metal in your mouth. And then when you put the wine in your mouth, you get these oxidation or reduction kinds of reactions happening. And sometimes for some people that can be unpleasant. And so there is some chemistry and some interesting interactions that happen that we can study. But there are also great wines that go with fish. And so, you know, again, it's really what an individual likes.
Okay.
I realized that I didn't turn my overhead light on. I'm going to turn the light on because it's really getting dark in here, and I'll be right back.
No problem. We can see you just fine, but I know the feeling. Well, so Harold, maybe you can answer this question. From what both of you talked about, it sounds like when something smells like something else, that's because there are molecular similarities or molecules in common. So for example, when a rosé smells like strawberries, that's because they share certain compounds or certain molecules. So is that correct?
Yeah, that's right. And I should have mentioned that when I started. I found my way from writing just about the smells of food and drink to the smells of the world, in part for exactly that reason, that, you know, I would taste a green tea and it would smell like seaweed or the seaside.
Mm-hmm.
And in wines, people often talk about, you know, sweaty saddle notes and floral notes, but there are no flowers in the wine. There's, I hope, no saddle. So that got me really interested in, you know, what is it in the saddles and the ocean that are being echoed by food and drink. And it turns out that what you said is exactly right. They share these fragments of the larger molecules that make them up. And so we have these echoes that go back and forth across the different realms of the world.
Sense. So Sue, kind of a slightly different type of question is about decanting. So of the chemicals you showed us, how does decanting affect that? Do they off gas? Are they oxidizing? What is going on with that process?
Well, most wines that are made now really don't need to be decanted. Some of that is historical. Before wines were filtered a lot, and so some of the decanting was to let particulate matter sort of settle out, and so then you could just have the liquid part and not the particulate matter. There are changes that happen as you open a bottle, as you pour wine into a glass, because the different compounds have different volatilities, they have different boiling points. And so we've done some work looking at the changes in volatility of a wine as you put it into a glass. I had a graduate student, Greg Harrison, look at that a few years ago. And so we see some compounds that are really volatile. You start to perceive them right away. And some of those are things like the hydrogen sulfide I mentioned, and Harold mentioned these primordial kind of outer space kinds of smells. And sulfur dioxide, which is formed naturally in fermentation, but also we add it as a preservative. And so those are really volatile. And so if those are in high concentration, pouring the wine, letting it sit, rest, and be exposed to air for a little while, you can lose a lot of that sulfur dioxide, hydrogen sulfide kind of smell. And then when you come back to it, you start to smell the other things that have maybe a little bit lower volatility. And so that's really what's happening. And so most of the time now wines are well made, and so they don't have a lot of hydrogen sulfide, they don't have a lot of SO2 to them. And so we're not seeing this big off-gassing usually, but we can see these subtle changes happening over time as different compounds start to volatilize, maybe as the wine warms up, as air flows over the surface of a glass. So that's what's happening.
Okay, thank you. So I have another question that's really for both of you, but I'll start with Harold. And you talked a lot about terroir and the role of rocks in wine flavors, but we're curious about soil. And there's a lot of buzz right now about the microbiome and its effect on grapes and taste. So could you talk a little bit about the soil part of the terroir?
I guess I really can't because except to say that it is important, and we're only just beginning to appreciate how important it is. I mean, we've known for a long time that the drainage of a soil, the availability of water, the availability of air, the availability of nutrients of various kinds, all of these do affect the way the grapevine kind of decides how much of its resources to put into the vine itself, how much into the leaves, and how much into the fruit, and which things go into the fruit. And so, of course, that's going to affect the nature of the wine that results. When it comes to the microbiome, that's our understanding that that's important is very, very recent. And I would say, Sue can correct me, but maybe the last 5 years or so that it's really become clear that that makes a tremendous difference. And I think we're still figuring out, just as we are with the human microbiome, we're still figuring out, because it is such a complex system, exactly what's going on. But your question made me realize that I left out a really important point in my discussion of minerality in Chasselas wines. I did point out that it was through the study of excrement that we discovered that these particular sulfur compounds were a part of the story. What I didn't mention was that those sulfur compounds have nothing to do with the soil. Those sulfur compounds are generated by, again, a complex interaction among the yeasts and their metabolism, the addition of sulfur in the vineyard and in the fermentation process, and trace metal contaminants from the equipment that was used to make the wine. In other words, it's the same molecules, but they come from a completely different source than they come from when you hit two pieces of flint together.
Aha, okay. Sue, do you have anything to add to that?
Nope, it's just very complex. The microorganism, we can't have wine without microorganisms, of course. We have to have yeast, and then sometimes we have bacteria, malolactic bacteria. we can use cultures to control that microflora, and we can have natural yeasts and microorganisms coming from the vineyard, from the winery, all the things that Harold's talking about. And so it's just really complex. The microbiome in the soil is influenced by so many things, and how that interacts then with the rootstock, with the vine, with other nutrients that you're adding to the soil, how the wine is made, all of that becomes very complex, and lots still to learn.
No doubt. Okay, well, let's move on then from wine to food for a couple of minutes. We've gotten quite a few related questions about how much of our perception of smell is innate versus a product of culture, lived experience. And I know, Harold, this is the topic of your book. So for example, would growing up on a farm affect one's sense of smell, just having been exposed to those particular smells? Are there innate reflexes like disgust, or does that also come from our cultural training? Could you talk about that a little bit, Harold?
Well, I could by saying that I deliberately chose not to talk about that aspect of things in my book, simply because it is so elusive. I mean, there are two different things going on. You know, there are the molecules that are emitted by the things in the world that we respond to, and then there's our response to them. And our response to them is determined entirely by who we are and how we're prepared to information that molecule is present. So it has to do with whether we've come across it before. If we have, under what circumstances do we associate it with something desirable or something not so desirable? On the question of innate disgust and things like that, even that seems to be, I mean, we do have innate preferences in taste. Sweetness is something that newborns gravitate toward. They don't need any training whatsoever. But very young children, of course, anyone who's had young children will know from experience, will put anything in their mouths. And that's just, you know, part of learning what the world is all about. And depending on the lesson you get when you put that particular thing in your mouth, you're either going to register it and put it in your database as something to be sought after or something to be avoided. So yeah, it too is a complex system that we've got in here, just like the vineyard is a complex system. And but the general point, I think, is that it's a very flexible system and adaptable.
Yeah, I mean, you have an anecdote in the book about ants versus lemongrass that just shows that the same smell can trigger very different responses from people. And Sue, did you want to add anything to that before we go on?
No, I think there's a lot of differences in the way, you know, I'm a mom, so I've done this, you know, with a kid growing up, that we spend a lot of time reading books with kids and talking about colors and teaching kids colors and teaching kids sounds, but we really don't spend a lot of time teaching kids aromas. And I think if we did, we would probably all be much better at recognizing and describing aromas in the world around us. If I spent, you know, as much time on a walk with my son saying, oh, did you smell that? What does that smell like to you? This is the language I use. We would all be much better at it because we start out very young, starting to gain that experience and the vocabulary. So that's all I would add.
Thank you.
And I would add something on top of that because Sue reminds me of this. So I have a couple of kids, and we weren't programmatic about it, but once I started focusing on this aspect of things, I did, and we did sort of interrogate each other. You know, we'd be walking down the street and I'd say, do you smell that? What do you think that is? Or eating something that we haven't eaten before, or so it's something that we can not only do as an educational process when our children are young. It's something we can do throughout life simply by comparing notes with people. I mean, that's one of the things about smell, is that it's such a private sense, you know. You can't point and say, you see that over there? You have to kind of sniff and say, can you smell that? It's a very different experience, but I think a really wonderful one to share.
Yeah, yeah, that's true. Well, with just a couple of minutes remaining, I'm going to close with a question for both of you, which is what's next? So I'd like to start with Sue and ask you, what is the research agenda for the next decade that you can tell us about?
Well, I'm going to kind of go two different areas, one on the analytical chemist side of me. There are just still so many things in our world that we don't know what the molecules are that contribute to the aroma. And so there's lots of things that we can still study to learn more about their complexity and what the compounds are. I mean, I think these di and trisulfanes is a great example. Just in the last 5 years, we started to understand and be able to identify and understand their contributions to aromas. So that's on the analytical chemistry side. On more the neurobiology, cognitive science, brain processing kind of side, which is not really the work that I do at all. To me, that's an area that is really still so unexplored, but is exploding. Just this whole understanding of, do we perceive things as an aroma object, like this is wine and I perceive it as wine? And how do I do that compared to being able to perceive the elements of that wine aroma, that bell pepper character or that fruity character? And so that's kind of a new area that people are really just now starting to really think about and study and really learn more about the processes of how we perceive the world around us.
Great, thank you. Harold, what's next for you?
Well, having spent 10 years on this book, I'm taking a little break right now. But you can count on seeing me up at UC Davis shortly, because my book on food and cooking is approaching its 40th anniversary. I published for a new edition for the 20th, and so I think I'm due. So I'm going to have to come up and catch up on what Sue has been doing and what her colleagues have been doing and redo my book.
Wow, well, we all look forward to that. Okay, so I have to stop the questions now. We had way more than we could ask you, but thank you so much for taking this time. And I'm going to turn it back over to Andy now. I think. Yes, there he is.
Well, thank you very much. Thank you, Sue and Harold, for such a fascinating discussion this evening. And also I'd like to thank Mackenzie for moderating, keeping us on track. I'd also like to thank the Robert Mondavi Institute and the library staff for all the work they've done preparing for this evening's program. And finally, I'd like to thank all of you for attending. We'll post a recording of this evening's program in a few days, so you can share with your friends, family, or colleagues who might be interested. And we hope to see you at a future Savor event. And as you leave, please take the opportunity to provide feedback and suggestions for future events. And good night.